This is our plate resulting from the ligation and transformation lab. A plus means ampicillin resistance. It was treated with xgal. T means that it is our transformed bacteria. Inside we can see two types of colonies, blue colonies and white colonies. A blue colony then indicates that ligation was not successful. In other words, it does not have the segment of DNA responsible for degrading estrogen inside. The white colonies are the ones that we want because the ligation was successful in those. Now I'm going to move to the next step using these white colonies. With this procedure, I'm going to take the colonies from the transformed bacteria with the ligated vector, and I basically want to grow them on Luria broth. So I'm going to start by using the Bunsen burners. We'll turn this on. We barely hear gas. And we'll light that Bunsen burner. I'm going to take my loop and flame my loop so it's nice and hot. Then I'm going to go here, take my bacteria. I just want one of the white colonies, a little part of one of the white colonies, so take a little bit out there. Then I'm going to take my tube, flame the tip, put the bacteria in, shake them all around, flame the tube afterward, put the lid on, put it back, and flame my loop. Okay, we'll turn off the gas. Now I'm going to take this tube then and put it into the incubator for 24 hours. 24 hours have passed. Here's my bacteria that has grown. We can see that it's all cloudy in here. These are my transformed and ligated E. coli. My first step is going to be putting 50 microliters with my pipetter onto two plates. One is a plate with ampicillin positive, one is a plate that is ampicillin negative. Just get my tube right here, and then we'll just go up, get 50 microliters. I'm going to mix it all around, make sure that all my bacteria are fully mixed in there. We'll then put 50 microliters onto this plate. Switch pipette tips. We'll get another 50 microliters. Shoot those on this plate. I'm going to light the Bunsen burner. So I turn gas barely on until I barely hear it. We'll light that. I'm going to heat my loop. Then I'm going to just cool it down on the edge over here. And I'm just going to take those bacteria and get them throughout the entire plate. Flame my loop. Cool it off on the edge. And then we'll just take those bacteria and get them through the entire plate. Flame my loop at the end, make sure that it's sterile. Now I'm going to go in and incubate these two plates in an upside down manner then for 48 hours. Alright, 48 hours have passed. I have my two plates. This is my minus plate, no ampicillin, and I labeled these before I put them in the incubator. No xgal, these are my T for transformed bacteria. Positive, meaning ampicillin, has xgal, and T for transformed. So for you, you want to go to your lab report, look at the table, and you want to fill in your observed values for the table for growth on both of these plates and for color. And color-wise, I don't see any blue colonies at all all of these appear to be white and growing. Now it's time to do the transformation. Here I have a tube on ice. This tube has my T vector. This T vector is what you obtained online when you went in and worked through the lab report. The T vector then has a vector with the CRISPR Cas9 complex with a signal RNA that is going to allow you to knock out ampicillin resistance gene. Here is our C vector, which is our control vector. It basically has a CRISPR Cas9 complex with a randomized signal RNA that will not knock out the ampicillin resistance gene. Here then we have an empty centrifuge tube or a sterile microcentrifuge tube labeled C for control. This one is labeled T for treatment. So I'm going to start by adding ice cold calcium chloride. We want the biggest volume first, so we have 250 microliters of our cold calcium chloride. Cold calcium chloride then, as you recall from the transformation lab, allows for the cell membrane to be compromised so that the bacterial cells will take in the vectors. So I'm going to add this ice cold calcium chloride to each of these tubes. Next step then, I want to add the E. coli. So I want to add 50 microliters of E. coli to both of the tubes. So here's my E. coli. It's cloudy throughout the whole thing, so that's a good sign that we have bacteria everywhere in there. I'm going to then take 50 microliters out. I will put it in our control tube. I will take another 50 microliters. And put it into our treatment tube. The next step then is to add the vector. So I need a third type of pipette tip, so I have this set on 10 microliters. So I'm going to take this out here, and I will take 10 microliters of my T vector, place them into the T tube, and 10 microliters of my C vector. And place them into my C tube. The next step is to let them sit on ice so those plasmids or vectors can get into the bacteria. So we're going to put these on ice then, and we'll let them sit there for 15 minutes. Okay, 15 minutes have passed. I now have a dry bath here. This dry bath is set to 42 degrees centigrade. So I'm going to take both of my tubes then, and I'm going to put them into the dry bath at 42 degrees C, and I'm going to leave those for 90 seconds. All right, 90 seconds is up. So I will take these out, and then I'm going to place them back into the ice then for 2 minutes. All right, my 2 minutes is up now. I want to take 250 microliters of urea broth and put it into both my C and my T tubes. So I have 50 microliters of sterile urea broth into my C tube, and 50 microliters of my urea broth into my T tube, and I'm going to let those sit for 15 minutes at room temperature. 15 minutes have passed. I'm now ready to plate my bacteria that have been transformed with the T vector and the C vector. So I have a ampicillin plate for the C vector labeled C. I have an ampicillin plate for the T vector labeled T. I'm now going to go in and just take my pipetter. I want 50 microliters. So I will start by getting a new pipette tip. I will take my T into my pipetter, make sure I put it on the right plate. Get rid of the tip, replace that tip. Go ahead and get C. And then put my C on the C plate. I need to then spread that around. It has bacteria on it, so I want to make sure it's nice and sterile. Turn my Bunsen burner on until I barely hear the gas. And I need to flame my loop. And I'll just cool it down gently on the side, keeping my plate fairly near that Bunsen burner. And we'll just spread this out evenly throughout the entire plate. So it's nice and evenly distributed all the way across that entire plate. Flame my loop, make sure I do not get any contamination whatsoever. And we will spread this one out. Oops, cool it off a little bit. Spread this one out through the entire plate. Making sure I didn't lose any of the bacteria on that plate. Reframe my loop to kill all of the bacteria then. And I'll consolidate these plates, flip them upside down, and incubate them from between 24 to 48 hours. Here are our two plates after 48 hours of incubation. We have our C plate here, we have our T plate over here. Each of these dots is a colony. We have bigger colonies over here because there are fewer bacteria on the plate, so they grew bigger. Smaller colonies here because with more bacteria they grow less. Your job now is to count all of those individual dots or colonies. Here is a cluster of colonies. You have to do your best in delineating out those colonies. Once you count the colonies, then you want to make a data table in your lab report, record your data in the data table, then get onto Canvas and add your data to the bigger data set, and then analyze the data for the whole class.